invited to give a talk here. I'd like to share with you my research findings, and different from the previous two speakers is about the technical issue. The topic is exploration in chemical and biological analysis of carbopolysaccharide. And I will go through the challenge in the analysis and what we have done to address these concerns. First of all, why polysaccharides? I would like to draw attention to the importance of polysaccharides using two examples. The first one is one of the mostly used herbal medicine, ashwagandha radix. That is a cheap tonic of herbs. And we, in a study of the quality control, we find that the small molecules found in these herbs may not be the chemical ingredients responsible for its functional, its traditional function. Because they feel reported to show the immune enhancing activity. And we analyzed the breeds and found that the bigger roots contains less the small molecules. And the traditional clinical experience told us the bigger the better. So in other words, these small molecules may not be the real active ingredients for the traditional function of the cheap tonic. The bigger roots contains more polysaccharide, and polysaccharides have been well known to show the immune enhancing activity. So we have known the polysaccharides might be the real active ingredients to explain the traditional function as cheap tonic herbs. Another example is the famous peach wine 906 that is the herbal botanical drug application case. And the team that was the inventor found that without the jujube, this formula will lose its function in the golden index of the cancer therapy, that is the prevention of mortality. Jujube, what's jujube? More than 90% of this herb is saccharides. In other words, saccharides is responsible for the golden index of the cancer therapy of this formula. So that's how important the polysaccharide could be. But the fact is, few medicines, few drugs are based on polysaccharides. Why? There are three difficulties. One, the poor quality control, and the poor structural analysis. The macromolecules, the chemical structure could be very complicated. And the final one is poor bioavailability. If the polysaccharide cannot directly contact the cells, people will believe it's a bioactivity, right? And if the polysaccharide cannot contact the cells, what's the meaning of the in vitro study? So we spent decades to address these concerns. The first one, the difficulty. Why does the difficulty in the quality control? In other words, the qualitative and quantitative analysis of these chemicals. It's because these kind of chemicals, they don't have the UV absorption. They are hard to ionize, so we can't use MS to detect them. And they are highly polar macromolecules, so it's hard to separate them by the commonly used chromatography. So normally we know the LCMS, but it's weak to detect and analyze the polysaccharides. And other conventional methods are also weak to detect the polymer. For the bioactivities, for the chemical structures we can find, we need a lot of complicated method to analyze the chemical structure. So that made the chemical structure results could be poorly reproducible. So that is a fact. Nobody reported their polysaccharides that had been reported by other research people. Even for the same herbs, there are hundreds of reports, but no one states everybody reported new structures. The reason is the chemical method we use. In other way, we found that actually the different batches of the same herbs, they show the highly consistent chemical profile of the polysaccharides. So that's the concern. How we can accurately describe the chemical structure of the polysaccharides. And we did our efforts, and we use two cases to report it. The first case is the Dandobi officer. And we use the chromatography, that is a method to analyze the molecular size distribution of the water decoction. And we found that in the water decoction of this expensive herb, we find that's DOP, the polysaccharide fraction, that is highly specific to the authentic species. In other Dandobi species, there's no this marker. So we developed the first polysaccharide marker to address the qualitative and quantitative analysis of the quality of these Dandobi herbs. And this approach has been successfully used another medicine, that is cordyceps, very expensive, right? So the authentication is very important. And we developed a similar polysaccharide marker. It's specific to the caterpillar part of the authentic species. So we can use this marker to do the qualitative and quantitative analysis of the quality of the cordyceps. But that's a concern, because at the same time, many herbs, they don't have any polysaccharide markers once they mix with the other materials. So what we can do? And we have an idea. Because the saccharide could be divided into three tiers. One is a monosaccharide, then oligosaccharide and polysaccharide. So if we can't use polysaccharide markers, maybe oligosaccharide. Compared to the polymers, oligomers demand some specific chemical structure, chemical features, right? And compared to the monosaccharide, they are similar. They are also the small molecules and could be analyzed by the conventional method. So we tested these ideas and successfully established the oligosaccharide markers for the Dandobi officer methods. And this approach also successfully developed to find the oligosaccharide markers for the individual polysaccharide herb formula, There are two herb polysaccharides, and we developed the individual oligosaccharide markers to analyze the polysaccharides in this formula. And this method has been invented in terms of patents, and these patents help us to establish the company to provide a commercial service to the industry. And the next difficulty is chemical structure analysis. And when we studied the Dandobi officinal polysaccharide marker, we analyzed the chemical structure using the conventional method, and we found that is a black bar containing both mannose and glucose. But when we changed to the oligosaccharide approach, we found that the main black bar doesn't contain any glucose, and the glucose could be removed by the discharge operation. So we found that actually the polysaccharide of the Dandobi is a mixture of the starch and the mannose. So based on these cases, we developed a new criteria for the polysaccharide purity. We know polysaccharide naturally exists in the herbs, right? And the people using the artificial method to separate them. If success, we can get the separate chemical structure. If not, we will get a mixture. And the chemical structure could be much more complicated. So for these different chemical structures, linked or mixed, that's the question. Now we developed the enzyme diagnosis purity criteria to test if we get a pure enough polysaccharides. And this is the conception. This is based on the monosaccharide analysis, and we can find what kind of saccharide it contains, and then we can choose the enzymes. And if we use enzyme to catalyze the target polysaccharide, and we find the molecular size changed greatly. That means all the polysaccharide could be degraded by the enzyme. That means that is pure enough chemicals. But if only a part of the polymers could be hydrolyzed and there's a significant residue, that means that's the mixture. So we can use this method to determine whether or not we get a pure enough polysaccharide. And furthermore, we can use this method to test what kind of polymer structures the mixture contains. So this concept has been tested by the model studies. We use a different model. This is the dextrin and the polona at the same molecular size, and we can find, we know they are mixture because we made it. But if we treat it as a pure chemical, and the chemical structure could be much complicated. That's another model using the reference standard. And the different reference, this is neutral saccharides and acidic saccharides and the different compositions. The result confirmed that this purity criteria based on the enzyme analysis is efficient to figure out the mixture. This is the real cases. We analyze the chemical structure of the mushroom polysaccharide. Using our method, we find that, okay, the usually reported polysaccharide actually is a mixture. This is the result. It's a mixture of starch and the beta-1,3-glucan and the alpha-1,6-galactan. So that's tell us the natural polysaccharide may be just a simple mixture of much simpler polymers. So the chemical structure analysis difficulty could be easily solved. So the last concern, the poor bioavailability of the oral polysaccharide. And we try our best to figure out the mechanism, how the oral polysaccharide works in our body. And we find the first one, they cannot be absorbed into the blood and they cannot be digested by our stomach. But when they encounter the gut microbiota and could be quickly degraded into the short-chain fatty acids, and simultaneously they can regulate the composition of the gut microbiota and their functions, and then show some activity like the anti-cancer. So that is one way. And we even figure out what kinds of bacteria degrading the polysaccharide. But that's not enough, because if all the polysaccharide follows the same way of the gut microbiota, why they not show the diverse activities? And here I would like to use the second example to show what we find a very interesting bioavailability pathway. The case is that is again. In our preliminary study, we found that the radical polysaccharide, for short, the RAP, it can protect the chemotherapy supplies for bone marrow. That is the in vitro study. And for the in vitro, we find that the RAP can directly contact the stem cell of the bone marrow from the chemotherapy. So that's in vivo and in vitro, which indicate the same direction. The problem is, can they contact together? Can the polysaccharide contact the stem cell? We directly detect the polysaccharide in the bone marrow, and we find that the polysaccharide really co-located with the stem cell and the dendritic cell in the bone marrow. That is what we find in the animal samples. So there's a hypothesis. There must be one unknown pathway for the polysaccharide to enter the body and contact with the cells, but we don't know what it is. So we start our tracking. First of all, we need to know the RAP's activity rely on the immune system. So we confirmed this and use the tumor model. We found that on the normal mice, the biopsy mice model with bearing tumor, the RAP can show the significant anti-cancer effects. But when it changes to the new mice, that is immune deficient mice model, the anti-cancer effects disappear. And we can find the similar effects in terms of the immune cells. So that tells us one thing. The RAP's activity really rely on the immune system. That's one point. Another point is to find during our tracking. We use a different method to track the RAP dosed to the mice after the first hour. And we find that using the color reaction to indicate the sugar property, we found that after 3 hours, we dose the polysaccharide, and the polysaccharide still shows the sugar properties in our small intestine. But once it goes to the large intestine, the sugar property disappeared. That means the RAP are not a sugar anymore. And for the inflorescence, we find the similar result. And once again, we use the molecular science distribution method to detect whether or not the polymer has been degraded. And we find that in the small molecule, in the small intestine, the RAP remains intact. But once it goes to the large intestine, it degraded quickly. So the small intestine is the second point. So we have 2 points. One is the immune system. Another one is small intestine. Small intestine is the last location RAP remains a polymer. When these two come together, we have one new word, that is intestinal immune organ. That is a pair patch. So we further detect the pair patch and find the RAP really enter the pair patch. And then we further go to the next lymph node, that's MLN, and we also find the RAP. And simultaneously, the RAP triggered the immune response in the pair patch. And we further detect the RAP directly target the dendritic cells in the pair patch. So we find that there may be a new pathway, that is intestinal lymph absorption pathway for the polysaccharide to enter our body to direct contact with the immune cells. And the next step is to identify the transport cell, because we found that this pathway is highly specific, only RAP. Another polysaccharide, the DOP, the dendritic polysaccharide, the dendrobiopsinal polysaccharide cannot. So let's go to the key evidence. We found that the green labeled polysaccharide IEP goes through the red labeled M cell on the parapath, enter the parapath, and then direct contact with yellow labeled dendritic cell. That's the moment we spend half a year to catch it. And this pathway has been verified on the human subject. So that's our conclusion. We find mice and human common pathway for the polysaccharide to enter our body, especially the lymphocyte route to contact the immune cell directly. And then we find that the trigger is DCB axis to produce the anticolytis IgA. And further, we demonstrate a dynamic map of the lymph-absorbed IEP. And we find it can go to every lymph node and enter the main organs like the liver, lung, kidney. So that's a bridge between the in vitro and the in vivo studies. Yes, this is the solutions we proposed to the three major concerns of the polysaccharide research. We developed the oligosaccharide approach and the enzyme diagnosis with the purity criteria, and then we developed the polysaccharide and oligosaccharide markers for the qualitative and quantitative analysis. And we also developed the third one, besides the gamma microbiota and the blood absorption, the third one is the lymph absorption, the available pathway for the oral polysaccharides. That's all. Thanks for the funding support and thank you for your attention.
Thank you very much, Professor.